Welcome everyone, this is Dr. Shen. I'm one of the instructors for Bio 372 Cell Biochemistry. And the other two instructors are Dr. Esposito and Professor Kaluski. So for the Bio 370 lecture, this lecture will be delivered by Dr. Esposito and myself. And the laboratory instructor will be the Professor Kaluski. And this course will be delivered online synchronously. Although it will be online synchronously, and we will post all the class video on the Blackboard. So it is very important that you will need to come to the Blackboard and then also to review those all the class videos. And before we continue the class, first of all, I'm going to go through the syllabus with you so that you have some idea about the class and the class materials and how we evaluate your grade. And before I share my screen to show you the syllabus, I want to show this is the book that we're going to use. This book, Biochemistry, that we're going to use for this class. Okay, so I hope all of you will get the textbook. And now let me share my screen, and so we can go through the syllabus. So this is the syllabus for our class, Bio 372 Cell Biochemistry. As I mentioned earlier, the lecture instructor will be both Dr. Esposito and myself, and this will be our email, and you are always welcome to email us if you have any question. And the lecture meeting time will be online asynchronous, and laboratory instructor will be Professor George Kaluski, and this is his email, and of course it's also online asynchronous, and you are also welcome to email him if you have any question about the laboratory. And the textbook will be the Biochemistry S edition. It's by the Campbell and the Farrell by the Cengage Learning, which is this textbook. Okay, so it is important that everyone should get a copy of the textbook. And then, of course, the laboratory, and then the Professor Kaluski will also perform some exercise from this book, the experiments in biochemistry, a hands-on approach. Okay, and then for the evaluation, for this class we will have 4 exams. Okay, we have exam 1, 2, and 3. Each one will be 15% of your final grade, and then the final exam that'll be cumulative, that'll be 33% of your final grade, and the laboratory will be 22% of your final grade. Okay, so that'll be evaluation for this class. And of course, as you know, that we will perform all our exam online. Therefore, it is very important that you have to follow the academic honesty procedure. So each student is responsible for being aware of what constitutes cheating and plagiarism, and for avoiding both. And very important, cheating of any kind will result in immediate failure of this course. So although we will perform the online exam, we will give you this exam on the Blackboard, and this Blackboard exam, they will only show 1 question at a time and no backtracking. And also it will be you are required to complete the exam at a certain period of time. So if you try to for example, if you try to backtrack to go back the previous page, then you'll be logged out from the Blackboard, you'll be kicked out from the exam. So that's why it is very important from the beginning. I have to tell you, you have to realize all our exam will be, the question will be shown 1 at a time and no backtracking, and all the questions will be randomized so that we will make sure that the exam, the test will be fair to everyone. Okay, and then here will be the office hour for myself. For myself, so basically I will give you one Blackboard office hour between 11:30 and 12:30. So you have to come to the Blackboard, the Ultra, okay? And then I'll be there. I'll be available on the Blackboard, and also January the 10th between 11:30 and the 12:30. I'll give you the in-person office hour. So you are welcome to come to my office at 6S143 if you have any question. Of course, beyond these hours, you are always welcome to email me. And then, when you email me, remember to put the BIOS 372, your last name in the subject line, so that I know it's from the class. Okay, and so that I will be able to respond to your questions. And then for Dr. Esposito, and he'll be her statement and to perform the office hour. So you have to go through these, all right? And then to set up this the Remind School messaging app. Okay, if you have any question about to set up the app, please email her so that she can guide you through to set up the app. Okay, so that'll be interactive office hour between Dr. Esposito and you. Okay, so that will be the office hour and the evaluation and also academic honesty statement. And then here will be just the syllabus for the lecture. So in here that will be only for lecture, and then of course Professor Kaluski, he will provide the laboratory schedule, so you will follow his schedule. Okay, so in here basically in the first column will be the date of the class. All right, so like for example, first one will be January the 3rd, and the second column will be the chapter that you will need to study from the class video. And then, of course, here will be the topics from those chapters. And then the last column here will show you which professor is teaching that particular topics. So, for example, I will be teaching the from chapter 1 through chapter 5, and also from chapter 9 through chapter 13, plus chapter 15 and 16. So I will be responsible for all these chapters, all these topics. Okay, and then Dr. Esposito, she will then teach all the rest of the chapter of the book. Okay, and then as you can see that for the exam 1, we will perform the exam 1 on January the 10th at 4:30 p.m. All right, and that will cover chapter 1 through 5 and the chapter 9 through 11. So that'll be exam 1. And then January 14th, that'll be exam 2 at 4:30, all right, at 4:30. Remember, this will be the time that you need to come to the Blackboard to perform the exam, okay? Exam 1, exam 2, and then here will be the topic will be the topic on the exam 2. And then the exam 3 will be January 21st, okay, 4:30. And then this will be the topic that'll be on the exam 3. And then final exam that'll be on January 24th at the 4:30, okay, at 4:30. And then this will be cumulative exam, the whole books. So basically, this will be the general introduction to our syllabus, all the class materials, and how are we going to perform the class. Okay, if you have any question, you are welcome to email me or come to the office hour to talk to me. All right, so this is the syllabus. Then the next thing I want to show you is the Blackboard. Okay, the Blackboard. So basically, this is the Blackboard. So when you look into the BIOS 372 Blackboard, then you will see this, and then from the left, you will see the column, the syllabus. So I will post the syllabus here, and then Dr. Shen's lecture. So basically, all my class video will be under this tab, and then Dr. Esposito's lecture will be under this tab. Okay. So for example, if you click this, then you will go to the Dr. Esposito's folder. And then if you click Professor Kaluski's lab, then you will come to this folder here. So basically, he will give you this laboratory syllabus and also the Lobster and PowerPoint and quizzes. Okay, so you have to follow all his procedure to perform your laboratory exercise. And then for the lecture, so basically you will need to come to mine or Dr. Esposito to review those class video. And then of course, for the exam, then we will create another folder for all the exam. So you will need to come to the Blackboard to perform the exam 1, exam 2, exam 3, and the final exam. Okay. And then for my office hour, as I mentioned, I have the Blackboard office hour. Then you will need to come to collaborate. So you would need to come to collaborate, click this, and then come to collaborate and ultra, and then come to here, the section options, join the course room. Then I will be available in the course room here for the office hour. So basically, this will be the introduction about the Blackboard. So if you do have any question, and you are very welcome to email me or come to my office hour so we can discuss this further. Okay. Lecture. Okay, so the chapter 1, biochemistry and the organization of cells. So basically, this chapter is a pretty much a brief review of the chemistry and also the cells, right? Especially the cell structure. So it's like an overview of the chemistry that you learned before, and also the biology that you learned before, and then we try to make the connection. All right. So that's why we have this biochemistry. Okay. So for the next few slides will be the about the basic things and also the chemical foundations of biochemistry. Okay. So we know all the living things make use of the same types of a biomolecule and all use energy. So as a result, all living things can be studied using the method of chemistry and physics. And in terms of the biomolecule. So this term, biomolecule. So basically, biomolecule can be from a very simple molecule like water, methyl and carbon dioxide, ammonia, nitrogen, and also the hydrogen. And also it can be as big as like the carbohydrate. Okay, carbohydrate. All the living things they use the carbohydrate. Okay, the carbohydrate and the nucleic acid, lipid. All right, so these are the bigger biomolecules. So all these are the biomolecule that's shared by all the living things. All right, and that's the reason why we need to study the biochemistry. And then this is just the levels of the structural organization in the human body. All right, so from the smaller this like this small these atoms, right, with oxygen and hydrogen in the body, and also then water, right? We have a lot of water, right, in the bodies, and then the protein, and then the cell, right? So we have the nucleus, and some plant cell has the chloroplast, but in human we don't have the chloroplast, and then in the human body we have this mitochondria, Golgi, and the neuron, bone cell, and then finally you have this body system of organism. All right, so that's the level of the structural organization. If you break down these to the smallest one, that's the atom. And that's what we are going to review later as well, the atoms, okay? So the chemical foundation of biochemistry is the organic chemistry, because it's a study of the compound of carbon. And this is because all the cellular apparatus of living organisms is made of a carbon compound. So the carbon compound, the most obvious one would be the carbohydrate. That's a carbon compound, okay? And even the nucleic acid, right? Even the nucleic acid, like DNA, DNA, we have the nitrogenous base, and also we have the sugar, right? The sugar, and then phosphate. And the sugar, that's a carbon compound, all right? So, so that's why all the cellular apparatus of living organisms is made up of the carbon compound. And then biomolecule are part of the subject matter of organic chemistry. And the reaction of biomolecule can be described by the mass of organic chemistry. So that's the reason why the organic chemistry is important to the biomolecule. And the first experiment to demonstrate that inorganic chemical can synthesize we can synthesize those organic chemical in vitro through the inorganic chemicals. That's in 1828. This Waller experiment. So in this Waller experiment, basically it's the synthesis of urea from the ammonia cyanide. Basically, just heat the ammonia cyanide, and then you can produce the urea. So basically, so this is the concept of vitalism, all right? Basically, before the 1828, the vitalism basically said all the organic compound could be made only by the living system, but this Waller's synthesis showed the organic compound, which is the urea, all right, did not require a vitalistic explanation. It can be synthesized in vitro. Just obey the law of the chemistry and physics, and then you will be able to produce this urea. So that's the Waller's experiment to demonstrate that to synthesize the organic compound outside the living system. Okay. And then, as I mentioned, the organic chemistry is the foundation of the biochemistry. And so here's the functional group that you should know. These are the functional group biochemical importance. So like alkene, like this, the double bond here, all right? And alcohol, we have the hydroxy group, okay, hydroxy group, and then ethers, now we have ether group, and then amide group, amino group here, and thiol, that's a sulfide group, and then aldehyde, we have carbonyl group, all right? And ketone, that's a carbonyl group, like here, carbonyl group, and amide here, and phosphoric acid ester, the phosphoric ester group. So these are some important functional group, okay? And then for this periodic table, so basically just to have a brief review. So the period here, the first row here, okay, so let's just show the you have the 2 element, and then second row we have 8, 18, and then 32, right, elements. And group that we call as a family, that each column that represent the group of that's a family. And the same group of the family, they have the same amount of valence electron. That's what we would discuss later, okay? So so this is a periodic table. So the electron arrangement, so that would be the critical thing for us to understand how the atoms join together to form the compound So like the ionic bond, covalent bond, which we will discuss in the next few slides. So the electron configuration, that's the arrangement of electron in atoms, and valence electron, that's the outermost electron that involve the chemical bonding. And then, as I mentioned, the same family, right, the same group of the element, they will have same amount, same number of the valence electron. Okay, so like this column here, they have one valence electron. Okay, that means one electron in the outermost, right, shell, so that that participate the chemical bonding. And then this is just the electron configuration. So we have this different level. The S sublevel has one orbital, and then can hold the two electron. And then the P sublevel, all right, so has three orbital, all right, and each orbital is two. Each orbital is two electrons, so maximum you can have six electron. And then the D sublevel has five orbital, therefore you can accommodate 10 electron. All right, if we go back here, so so this column here, basically, they say, for example, this column, this family, all right, they have same number valence electron. So that means they normally they have these at the outermost shell, they have one electron in the valence shell. Okay. And then to write the electron configuration, the hydrogen has one electron, so it's 1s1, right? And the lithium has three electrons. See the outermost has one valence electron, is similar to this the hydrogen, all right? But the first level, the 1s orbital here, is fill up two electrons, so it's 1s2 and 2s1, all right? So that's how they distinguish. So this is 1s, so two electron, 1s, and 2s, these two electron here, and then 2p, the six electron, all right, the three orbital, 2p, and then 3s and 3p, 4s, 4p, and then 4d and 3d here, that 10 electron here. Okay, so that's the electron configuration and the periodic table. And the valence electron, which is the outermost electron in an atom, and this electron involved in bonding. Okay, and the noble gases are extremely stable. We call it inert because they don't readily bond to other elements. And the stability is due to a full complement of valence electron in the outermost S and the P sublevel. Okay, this S or the P, all right, you have to fulfill the complement here, S or P sublevel, right? And then we have this octet rule. So that means elements usually react in such a way as to attain the electron configuration of the noble gas. The noble gas, which is this column here, all right? This column, I give you the noble gas. This column here, all right? So, so basically, all these elements, they will try to attain the condition to the nearest noble gas condition, all right? So now the octet rule will be element on the right side of the table move right to the next noble gas. If on the left side, move backward to the noble gas of the previous row, all right? So on the right side, all right, on the right side here, they will move to the next noble gas. They will try to get the configuration to be the same as the next noble gas. If it's on the left side, all right, so for example, for the sodium and magnesium, they will try to move back to here, all right, to the previous noble gas here. Okay, so left side, you try to move back to the previous noble gas, and on the right side, move to the next noble gas. That's the octet rule. Okay, and then during this process, the atom will again lose or share electron in chemical reaction to attain this more stable energy state. Okay, and then question so far? Very good. Okay, so then the ions, all right? So now the ions, the electrically charged particle result from a gain or loss of one or more electron by the parent atom. So that'll give you an ion. So we have a cation, which is positively charged, all right? That's cation. Let me see. Positively charged. And so basically, those metallic elements tend to form a positive charged ion called a cation, result from the loss of electron. Okay, the example is here. All right, the sodium. All right, so lose one electron here, and then you become the positive here. All right, positive charge cation, and then anion, that's negative charge, result from the gain of the electron. Okay, because you gain this electron, so so it's a negative, right? Negative here. Okay, so it's negative. And the chemical bonding, the chemical bond, which is the force of attraction between any two atoms in a compound. That's the chemical bonding, all right? This is just the dot system, Lewis dot system, to show you the element, okay? So the dot system to show you this one valence electron and two, all right? You have 3, 4, 5, 6, 7, 8. Okay, this is the noble gas here, all right? And then for the element in this column, tend to lose this one valence electron, and this column tend to lose 2, all right? And this column here tend to obtain 1 extra electron, and this one to obtain 2 electron. Okay, and then the ionic bond, which is a transfer of 1 or more electron from one atom to another. Now will form the attraction. That form the attraction due to the opposite charge of the atom. Okay, we will explain that in the next slide. And covalent bond, basically, that will be the sharing of the electron between the atoms. So that will give you the covalent bond, all right? And ionic bond is a transfer of the electron. Covalent bond is a sharing of the electron. Okay, so in this one is ionic bonding, all right? So the sodium has a low ionization energy, readily lose this electron. Okay, this is electron from this Lewis dot symbol. Right here, the sodium tend to lose this 1 electron. Okay, the whole family here tend to lose 1 electron, and this family tend to lose 2 electron. Okay, So they will lose one electron, all right? And so become the positive. And then the chlorine here, chlorine has high electron affinity, all right? So they will gain this one extra electron, so it become the noble stable, all right? So that's the ionic bonding because the transfer of the electron from sodium to chlorine and to make the sodium chloride. And then for this covalent bond, so each atom attains a noble gas configuration by sharing the electrons. So like these two hydrogen, okay, each hydrogen, okay, they will share their electron with each other. Therefore, you have these two electrons for both hydrogen, and then they become stable. And this will be the covalent bond because they share this pair of the electron. Okay, so that's covalent bond. And the polar covalent bond, that's because for the covalent bond, we have to share the electron, but some element, they have even a stronger attraction than the other element. Therefore, the bond made up of unequally shared electron pair because at most of the time, the electron probably will be more closer to the one element than the other element. And that will give you the polar covalent bonding, all right? And this covalent bonding basically depends on the electronegativity. So that's the basically it's a measure of the ability of an atom to attract the electron in a chemical bond. So that's called electron negativity. So if you have a high electron negativity, then you have a greater ability to attract the electron than do the element with lower electron negativity. Okay, so basically in this corner, on the upper right corner, they have the strong electronegativity, okay? And on the lower left here, they have the least electronegativity. So that means if you share the electron with this element here, then the electron, so for example, the oxygen, the electron will spend most of the time near the oxygen element than for example, the potential, all right? Because 0.8 here and the oxygen is 3.5. That's electronegativity much higher. So electron will be more closer to oxygen than the potential. Okay. And then to name the compound and write the formula of compound, all right? So basically, there's two naming system, ionic compounds or the covalent compounds, right? So basically, you name the cation, all right? The cation, all right, the positive charged ion, followed by the name of the anion. And then a positive ion retain the name of the element. And then you change the anion suffix to the IDE. So for example, you have the sodium and chlorine. So you combine these two, you retain the name of the cation, sodium, all right? Sodium, and then the chlorine, all right? You have the anion chloro plus ID chloride, sodium chloride, okay? And then this one with sodium oxide, all right? And lithium sulfide, right? Calcium oxide. So you add the IDE to the end of the anion, all right? So that's how you name the ionic compound, all right? The ionic compound. Then if the cation, all right, of an element has several ions of different charges. As with the transition metals, you use a Roman numeral following the metal name. All right, so the Roman numeral gives the charge of the metal. All right, so like this iron chloride. All right, it's iron 3 chloride, iron 2 chloride, copper 2 oxide. All right, and you can use the IC to indicate the higher of the charge that ion might have, and use the OUS to indicate the lower of the charge that ion might have. So like this iron, chlorine, so you have ferrous chloride, ferrous, right? And then ferric chloride. This is higher of the charge, all right? This is higher of the charge, ferric, and lower of the charge, ferrous, okay? That's a common nomenclature system. Yes.
Interrupt you, but on the table, it doesn't look like it's FeCl2, but on the compound name over here, it says iron chloride.
Sorry, which one?
Over here, it says iron chloride, and it then says
iron chloride. This is compound name. Yes, iron chloride, iron 2, iron 3, and the compound name is this. Then then for the common name, so you have a ferrous, ferric. All right, so this is compound name iron 2, iron 3 chloride, and then the compound name that's ferrous, ferric chloride. Okay, it's the same for the copper 1 oxide, copper 2 oxide. That's a cuprous oxide, cupric oxide. So one is the compound name, one is the common name. All right. For the monatomic ions, you have a single charge ion. All right, so we have this hydrogen ion, and then hydride ion, lithium ion, fluoride ion. All right, so these are some of the common cation, monatomy cation and anion. All right. And then for the polyatomic ions, so the ions composed of 2 or more atoms bound together with an overall positive or negative charge. So within the ion itself, the atoms are bounded using a covalent bond. And the positive and negative ion will be bounded to each other with ionic bond. So this one is ammonia ion and sulfate ion. And this is just common polyatomic cation and the anion. And then for the covalent compound, all right, covalent compound are typically formed from the nonmetals, right? And so for the name of the element are written in the order in which they appear in the formula. And then also you have prefix indicate the number of each kind of atom, mono, di, tri, tetra, penta, to indicate the number of each kind of atom, okay? Like this one, so that's a carbon monoxide, right? And also we have the single bond, double bond, triple bond. So single bond, that's 1 pair of electrons are shared. Double bond, that's 2 pairs. And triple bond, that's 3 pairs of electrons are shared between 2 atoms, okay? And the bond energy, the amount of energy required to break a bond holding 2 atoms together. So a triple bond, the highest bonded energy, all right? And single bond is lowest bonded energy. And the bonded length, like distance, separating the nuclei of two adjacent atoms. So the single bond, all right, that's the longest bonded length, and triple bond is the shortest. Okay. So the organic compound. So basically, carbon form a stable covalent bond with other carbon atoms, right? And the carbon's atom form a stable bond with other elements like oxygen, nitrogen, sulfur, halogen. Okay. And carbon can form the single bond, double bond, or triple bond. Okay. So like this example, the triple bond, double bond. And then, and also you have many different ways to arrange the carbon and other atoms. All right. So organic basically have the covalent bond, and inorganic usually have ionic bonds. Okay. The organic molecule non-electrolyte. Inorganic three-dimensional crystal structure, often water soluble, dissociating into the ion electrolyte. And so basically, this is the table just to show you the summary between the organic compound and inorganic compound. All right. So the melting point, and so organic have lower melting point, right? And organic nonpolar, water insoluble. All right. And here's a summary. So this column is organic. This column is inorganic. All right. They have the same molecular mass. All right, organic compound has the covalent bond versus the ionic bond, and then the room temperature is a gas because the melting temperature is low. All right, and the boiling temperature is low. All right, and this is high and low melting temperature, high melting temperature, insoluble organic compound insoluble to water, and inorganic compound highly soluble in the water. Okay. And the hydrocarbons contain only the carbon and hydrogen. That's a hydrocarbon, carbon and hydrogen. They are non-polar molecules, so not soluble in water. Okay. And the soluble in typical non-polar organic solvents like a toluene, pentane. Okay. And the division, the family of the hydrocarbon. So hydrocarbon aromatic, you can see the ring, aromatic, aliphatic with alkanes, all right? The single bond, cycloalkane, you have the ring, a single bond, and then you have alkane, that's double bond, alkyne, that's triple bond, all right? That's the family of the hydrocarbon. And these alkanes basically only the carbon, hydrogen single bond, so saturated hydrocarbon, and alkane and alkyne unsaturated because you have the double bond, triple bond, and this bond can be broken and then to fill in more you can fill in with the hydrogen, for example. Okay. And the functional group, which we discussed in the beginning of the class, and the ATPs. So this is ATP. So how you form the adenine triphosphate, right? So you have this phosphate group, and then dehydration. So you form this anhydride of the phosphoric acid. Okay, phosphoric acid, and then you add another phosphate group, so you have the triphosphate, all right, adenine triphosphate, all right, so this is ATP. in the middle here, that's the sugar. Okay. So that's basically the overview of the chemistry, and then the beginning of the biology. So basically the Big Bang theory, which means you have this the result of explosion, right? So it's start to expand with great force, and then the temperature is very high, and then the average temperature of the universe decrease over time. And then the abundance of the important elements. So this is compared to the 1,000 carbon, right? 1,000 carbon, the abundance in the universe, abundance in the organism, the hydrogen compared to the carbon, and nitrogen, oxygen compared to the carbon in the organism and in the universe, to show all these important elements, all right? These are the biomolecules. As I mentioned in the beginning, these are simple biomolecule, and then you have more complex molecule like a protein, nucleic acid, lipid, all right? And these, the biomolecule, all right, could have arisen under the abiotic condition from the reactions of a simple compound, okay? So like in the Earth's ocean, surface of clay particle. So, and then this is the famous experiment from the standing mirror to demonstrate a biotic synthesis of organic compound. So you have, like, for example, the water. So that would be the like a sea, and then you heat the water, then the vapor goes through this atmosphere. You have meso, I mean the nitrogen amino group, and then hydrogen, and then during the thunderstorm, they mix together, and then they form this organic compound. Okay, and then when they cool, right, and then they form the organic molecule, and then drop to the earth. So basically, this is the Standing Miller's experiment, just to demonstrate the abiotic synthesis of organic compound. And then also this is the to show you the origin of the life. So for the biomolecule, okay, so it's also the complex biomolecule, which is also an important topic in the biochemistry, and we later on we will spend a lot of time to discuss. So that will include the carbohydrate, protein, nucleic acid, and lipid. All right, so these are four important large molecules that we will discuss in biochemistry. And the protein, nucleic acid play a role in life process. All right, so for example, this is nucleic acid. All right, so we have DNA, and DNA, as we all know, the DNA can be transcribed into the messenger RNA and then translate into these amino acids that give you the protein. Okay, and also we have these are the carbohydrate, and then that you can form the polysaccharide chain, all right, to get many okay polysaccharide. And this is just to show you the directionality in macromolecules. So we have these amino acid, all right, from the N terminal, all right, N terminal to C terminal, which we will discuss in detail in the chapter 2 and chapter 3. And so through the dehydration, all right, so you form this you form the polypeptide from the two amino acid, right? Through the dehydration. And then for this carbohydrate, also form this polysaccharide through the dehydration. Okay, there's a 1,4. So for example, this hydroxide and this hydroxide, so basically they will lose a molecule of water and then form this 1,4 link polysaccharide. Okay, And then this is the nucleotide. The nucleotide, all right, so from 5' to 3', all right, 5' to 3', all right, and nitrogenous base, sugar, phosphate, nitrogenous sugar, phosphate, all right, and through the dehydration, and then they form this nucleic acid, okay. And the enzyme and the genetic code. So as I mentioned, basically the enzyme, so it's a class of protein that display the catalytic activity. And the genetic code, that's a relationship between the nucleic sequence, nucleic acid, and the amino acid sequence. And now, as I mentioned earlier, so the DNA need to be transcribed, all right, transcribed into the messenger RNA, and then translate into the protein, all right. And during this process, you need the enzyme to help to expedite to to perform the activity. So during the transcription, you need the RNA polymerase, all right, and then for the translation, all right, you need a ribosome. So all these are protein, and then so that's why they say the chicken egg question. So what do you need to have to start this reaction, right? But recently they found the RNA, some certain type of RNA, they have a catalytic activity, and they are capable of catalyzing their own further processing RNA. Therefore, it could be the original coding material. Okay, those RNA, because you don't need to have the protein to process the activity. So this is a very important finding, that to show that some RNA have the catalytic activity, so they can proceed to perform the translation. Okay, that's called RNA world. So it just is you have you have RNA, they can have a different structure, and through the different structure, they can perform the translation to make the protein. Okay, so that's how this figure explain. All right, and so basically the next few slides, it's all explain how the RNA perform the this activity to start a life. Okay. And then the prokaryotic cell and eukaryotic cell, this is a review. Okay, the prokaryotic single-celled organism like bacteria, cyanobacteria, and the eukaryotic cell has a well-defined nucleus surrounded by a nuclear membrane. Okay, and can be single cells like yeast or paramecium. Okay. And the basic feature of all cells, okay, it doesn't matter it's a prokaryotic or eukaryotic cell, they all have plasma membrane, they have a cytosol, okay, that's a semi-fluid substance, cytosol, and they all have the chromosome and ribosome, okay, ribosome. So these are the basic features of all cell, right? Plasma membrane, cytosol, chromosome, and the ribosome. And the prokaryotic cell, no nucleus, right? And DNA in an unbounded region called a nucleus, and no membrane-bounded organelle, right? And the cytoplasm bounded by the plasma membrane. And the eukaryotic cell, DNA in the nucleus, right? It's bounded by the nuclear envelope, the membranous nuclear membrane, and the membrane-bounded organelle. So that's eukaryotic cell. This is a prokaryotic cell, animal cell, plant cell. The plant cell has an extra cell wall outside the cell membrane, and they have the membranes bound. They have the nucleus, all right, a nucleus, and there's no nucleus. And also membrane bound the organelle, all right? So the comparison, all right? So membrane, all right? Nucleus, no definite nucleus. Mitochondria, no mitochondria. Endoplasmic reticulum, no endoplasmic reticulum. Ribosome, right? Chloroplast, that's in plant cell, green plant cell, and no chloroplast. And that will give us these basic features for both eukaryotic and the prokaryotic cell. They both have plasma membrane, cytosol, chromosome, ribosome, right? This cytosol here, right? Cytosol. And we have this fire kingdom system, take into account different differences between the prokaryotic and eukaryotic, right? And we have three. The kingdoms are the Monera, Protista, Fungi, Planta, and Animalia. That's a 5 kingdoms. And 3 domains: Archaebacteria, Eubacteria, Eukarya. That's 3 domains. Okay, and the last 4th section of the chapter 1. Okay, so the biochemical energetics. Okay, all cells require energy to function. All right, and this energy, basically the energy comes from this light energy. All right, enter the ecosystem through the photosynthesis in the chloroplast. They will collect the light energy and then convert these carbon dioxide and water into the organic molecule and release the oxygen. All right, release oxygen, and then these organic molecule, all right, can go through this cellular respiration in mitochondria and then to release the energy. And this energy basically will power most of the cellular work, right? And then you lose the energy through the heat. Okay, and the photosynthetic organism, right? They will use the light energy to drive the energy requiring synthesis of the carbohydrate here. If it's a non-photosynthetic organism, they will consume these carbohydrate and use these as their energy sources. Then the energetic of a chemical reaction, basically, if the change in free energy is negative, the reaction is spontaneous, all right? Because you decrease the free energy. The energy change is negative. If the change is positive, that means you need to supply an external source, all right, to give you the positive change. So just like this the thermodynamic, right? So from high energy to a low energy, this reaction, right, will go down as spontaneous because that will release the free energy, and then the change is negative, all right? The change is negative. But if you want to go from this to opposite way from here to here, then you need to supply the energy because the free energy change will be positive. So the spontaneous means fast. So free energy of system, if the change is negative, right, spontaneous, it's asynchronic energy release, like this, right? From start to the end, you release the energy. And if it's a 0, that is equilibrium. If it's positive, non-spontaneous, it's endergonic. And then the change of energy basically would be the heat of a reaction, the change of a heat of reaction minus the temperature multiply the change of entropy, all right? The change of entropy. Therefore, if this change is positive, if this is positive and this change is, let's let's go to another more easier. All right, so here, from the first law of the thermodynamics, okay, the energy of the universe is constant, and that's called the law of the conservation of energy. All right, so the AV plus CD to AD plus CV. So basically, the if the reaction will occur, bond must break, and you break this bond require the energy, right? And so basically, if the so here you require the energy, and then when you form the bond, then you release the energy. And so basically, if the energy required to break the bond is less, if the energy required here is less than here, all right, then the energy release, there's a release of the energy, and then we call it exothermic reaction. Energy is a product of this reaction, okay? And if the energy required to break the bond, all right, is larger than the energy released, all right, then you need the external supply. That's called endothermic reaction, okay? So now the delta H, right, the heat, right? So exothermic reaction, A plus B, the reactant, to the C plus D, the product, right? The entropy, all right? So from this A plus B to C plus D, you release the heat, right? So the energy, right? So you have this, you plus the energy here. That's exothermic. And if you need to supply the energy here, so that'll be endothermic reaction, all right? You supply the energy to break the bond, okay? And then so the change in entropy, delta H, all right? Delta H, that's energy difference between the product and the reactant of a chemical reaction. So that be the delta H will be the delta H product minus delta H reactant, okay? Therefore, if energy is released in a reaction, the reaction exothermic, the delta H will be negative, right? That's the exothermic combustion of the metal here. So you release the energy, so it's negative. If the energy absorbed in a reaction, and then it's an endothermy, and this delta H will be positive, okay? So basically, exothermic reaction are spontaneous, all right? This is spontaneous because you release the energies, all right? Spontaneous, you release the energy. Endothermy are non-spontaneous because you need to supply the energy. Endothermy, you need to supply the energy. And then another factor is needed, it's called entropy, which is S, okay? That'll be the second law of the thermodynamic. So the universe spontaneously tends toward the increase in the disorder. That's entropy S. High entropy, that's high disorder. Okay, low entropy, well organized. Therefore, the entropy delta S will be S product minus S reactant. A positive mean increase in disorder, right? Negative, positive, so increase disorder, and negative will be a decrease in disorder, right? So like this from left to right, increase the disorder. So you have positive delta S, right? And also from solid to liquid, that's also a positive change of delta S, okay? So if exothermy and positive delta S, spontaneous, right? Exothermy. So as exothermy, so that means exothermy as delta H is negative, all right? Exothermy is delta H. So exothermy delta H is negative, all right? Delta H is negative here, all right? That's exothermy. And then delta S is positive. Then, so this is negative, this is positive, then the delta G would be negative, spontaneous, all right? Any question? Okay, so exothermy, all right, exothermy, delta H is negative, all right? And delta S, the exothermy, delta H negative, and the delta S is positive, therefore the delta G will be negative, then spontaneous, okay? That's how this explained here. That's what this means. Exothermy, all right, and positive delta S, spontaneous. If endothermy and negative delta S, then that'll be non-spontaneous. So if we go back to here, endothermy is a positive, all right? And if the delta S is negative, so if this is endothermy, which is positive, and this is positive, negative, all right, negative, then the total here will make the delta G positive. Therefore, it's non-spontaneous, all right? So exothermy is negative, all right? And positive, delta S positive, that will give you delta G negative. That's spontaneous. Endothermy, which is positive and negative, all right? Negative. So these two plus will be positive. So it will be positive non-spontaneous. Okay? So that's how this explain that equation. Okay, and for any other situation, depend on the relative size of delta H and delta S. Okay, and this just to demonstrate that increase the disorder, delta S is positive, all right, and this will be negative. And the free energy, the delta G equal to the delta H minus T delta S. T is in Kelvin. Okay. So for the negative delta G, the negative free energy, that will be always spontaneous. All right. So this is summary. So it's delta H is positive, delta S is negative, delta G always positive. Okay. So basically, delta H positive, positive, negative, then this must be positive. Okay, positive, negative, this must be positive. Negative, positive, then this will be negative. All right, negative, positive, that will give you negative delta G. Okay,